is Nushika binti Nurdi. Today, I would like to present our practical entitled Definition of Listeria monocytogenes. Listeria monocytogenes is a pathogenic foodborne bacterium that can cause listeriosis, which is a serious disease, especially among pregnant women, elderly individuals, and immunocompromised people. This bacterium can be found in contaminated food and environmental samples. Therefore, its detection is very important in food microbiology and food safety. In this practical, modified culture and biochemical method based on FDA and USDA protocol are used to detect and identify Listeria species. The objective of the practical are detect and isolate Listeria species from food or environmental samples using enrichment and selective media method. In addition, several biochemical and morphological tests are carried out to identify preceptive Listeria monocytogenes. Overall, this practical consists of 4 main stages, which are primary enrichment, selective enrichment, isolation, and identification. The first stage is primary enrichment using UVM1 booth. In this step, samples are incubated in a rich medium with selective properties to promote the growth of Listeria species while suppressing unwanted microorganisms. This medium also helps injured bacterial cells to recover and grow. Next is selective enrichment using Fraser-Brood. This medium contains selective agents such as lithium chloride and acriflavine that inhibit competing bacteria. Fraser-Brood also contains esculin and ferric ammonium citrate. When Listeria hydrolysis esculin, blackening occurs in the medium, indicating the preceptive presence of Listeria monocytogenes. After that, the samples are stricken into moss agar and pulcrum agar for the isolation stage. This selective differential media help isolate characteristic Listeria colonies, which usually appear as black colonies due to esculin hydrolysis. Finally, the identification stage is performed using TSIE agar gram staining, catalase test, and biohemolysis test to confirm the characteristic of preceptive Listeria monocytogenes.
Assalamualaikum and hello everyone. My name is Firdaus, S76211. So today, I will talk about method. This video presents our Listeria experiment using raw chicken and meat as the sample. First, the samples were transferred into F broth for enrichment to promote bacterial growth before further analysis. This step is important because bacteria may be present in low amount in the food samples. Next, the enriched samples were streaked onto agar plate using the streaking method to isolate bacteria colonies. The plates were then incubated for several days to allow bacterial growth. After bacterial growth was observed, gram staining was performed to examine the bacterial morphology and gram reaction under the microscope. In conclusion, this experiment involved enrichment, streaking, incubation, and gram staining for bacterial isolation and identification.
Now I will explain about the result of Listeria when streaking on pulcrum and TSAYE plate agar. When this sample is streaked on pulcrum agar, the results, and when incubated, the results form is black colonies, showing a positive control as the Listeria is present because the Listeria is able to hydrolyze the esculin on the pulcrum agar, forming black colonies. Then when the B sample is being streaked on TSAYE agar, the results form is white colonies when incubated, and this also shows a positive control because the white colonies is the morphological characteristics of Listeria, and the TSAYE agar has been used because this plate has the ability to isolate pure cultures. Then the results of Listeria has been further continued with gram staining by taking the culture from the TSAYE agar. Based on observation under microscopy with gram staining, when the magnification is 4x and 10x, the result obtained was as shown in the slide. Then this is the result observed in microscopy when the magnification is 100x. Then the overall result observed from the microscopy was showed that the bacteria is purple colored and rod in shape, and this suggests the presence of gram-positive bacteria and strongly suggests the bacteria is Listeria because Listeria is gram-positive and purple colored and rod in shape.
Okay, we move to the discussion. In this experiment, streaking was performed on pulcrum agar and TSI agar to isolate and identify Listerious bacteria. Based on the result of time, black colonies were observed on pulcrum agar, while white colonies were formed at TSI agar. This finding indicates the possible presence of Listeria species in the positive control sample. Palcum agar is a selective and differential medium commonly used for the isolation of Listeria spp. The formation of black colonies on Palcum agar occur due to the ability of Listeria to hydrolyze isculin, producing isolantine that react with ferric ion in the medium and form a black participate. These characteristics appear help differentiate Listeria from many other microorganisms. The selective agent present in Palcum agar also inhibit the growth of competing bacteria, allowing Listeria to grow more effectively. Meanwhile, on Tesai agar, what colonies were observed? Tesai agar is enriched medium that support the growth of Listeria and allow clear observation of colony morphology. The white to creamy colonies form are consistent with the typical appearance of Listeria monocytogenes on non-selective media. The growth on Tesai agar confirmed that the bacteria remain viable after isolation on selective media. The positive control used in this experiment successfully produced the expected colony characteristics on both media, indicating that the culturing, produce, and media preparation were performed correctly. The results obtained were consistent with the expected characteristics of Listeria spp, reported in microbiological study. However, several factors may affect the accuracy of the result, such as contamination during streaking, improper incubation temperature, or uneven streaking technique. In addition, colony morphology alone is not sufficient for complete confirmation of Listeria monocytogen. Further confirmatory tests such as gram, staining, and biochemical tests are necessary to accurate identification. Overall, the experiment successfully demonstrates the growth characteristics of Listeria on Palcum and Tesai agar, showing that selective and enriched media are important for the isolation and identification of foodborne pathogen. Okay, we move to the microscopic gram staining. Results show that the bacteria observed appear purple in color and rod shape under the microscope. The bacteria become more clearly visible and higher magnification, especially under 100 magnification. The purple coloration indicates that the bacteria were gram-positive because they retained the crystal violet stain during the gram staining procedure. This occurred due to the presence of the thick peptidoglycan layer in the bacterial cell wall. The rod-shaped area, also known as bacillus, is consistent with the typical morphology of Listeriosis bacteria. Listeria monocytogenes is commonly identified as a small gram-positive rod-shaped bacterium. Therefore, the microscopic observations support the previous finding obtained from the Palcum and Tesai agar, where characteristics colony of Listeria were observed. Gram staining is an important preliminary identification method because it helps different bacteria based on their cell wall structure. Gram-positive bacteria retain the primary stain, appear purple, while gram-negative bacteria lose the stain and appear pink after counter staining. In this experiment, the successful observation of purple rod-shaped bacteria indicated that the staining procedure was properly carried out. However, several errors could affect the accuracy of the result, such as over-decolorization, contamination, or preparation of the smear that is too thick. Those factors may lead to unclear bacterial morphology or incorrect staining result. Overall, the gram staining experiment successfully demonstrates the typical microscopic characteristic of Listeria spp, supporting the identification of the bacteria as gram-positive bacilli.
In conclusion, based on results from Palcum agar plate, the colony observed was suspected to be Listeria species because the colonies appeared small and black, as the Listeria tend to hydrolyze the esculin from the Palcum agar, forming black color. Besides that, the Palcum agar is selective medium supporting the growth of gram-positive Listeria by suppressing the growth of gram-negative bacteria and other competing gram-positive bacteria, allowing the Listeria to grow effectively in the Palcum agar. Then, based on the result obtained on the TSAYE agar, it was found that the colonies to be round, smooth, and creamy white in color, and this strongly suggests as Listeria species because these are the significant morphological characteristics of Listeria. For further confirmation, gram staining was done, and based on the gram staining results, it was found that the colonies to be appeared purple-colored and rod-shaped bacteria under the microscope, and this strongly proves that the organism is Listeria species, as the results obtained from the microscope is consistent with the characteristics of Listeria species. Overall, this experiment shows that the beef meat sample used to contain the Listeria species because the result from the Palcum agar, TSAYE plate morphology, and gram staining strongly supports the presence of Listeria.